Okay, guys, good afternoon. Today we will have our lecture on medical devices which are made of biomaterials and what is expected as a functional requirements, what types of biomaterials and the properties. Why biomaterials are special class of materials, because there are millions of materials, but not all materials are called biomaterials. So what makes them special and what are the qualifications they must have to call them as biomaterials? So that's basically the essence of today's lecture, okay? So, okay, I need to come out of this slideshow because I have a voiceover on the slide, so that's why you hear another voice. Okay, that was my voice because we use them for COVID time. Okay, so that's a most simplified way of saying what is a medical device. What we are saying is it's a medical product designed for the purpose of health care of humans. So any device that you are basically purposely designed for the better healthcare outcome of a human beings. So I made it very simple, whereas a week ago, when Dr. Joanne Lim, she presented a much more complicated way that is defined. And what she said to you is the right way. But we are engineers sometimes. We're just learning this course, but we really don't practice it. Then you just need to have a basic idea. Any device that is meant for healthcare outcome of human beings. So they include implants, prosthetics, drug delivery systems, diagnostic devices, tissue engineering, regenerative medicine. I think some of you, those doing PhD, some of you might be doing that kind of work. But in this year, I'm not presenting on tissue engineering lecture. We have a video lecture inside the files, but I think it's probably not needed. It's very specialized. If anyone doing research in tissue engineering or regenerative medicine, let me know. I can give you the access to the slides and presentation. And then imaging machines, like all the diagnostic imaging machines, X-ray machines, MRI, and so on. And any healthcare tools and instruments. So there are plenty of tools. You find them with a GP, general practitioner, or you go to a hospital, you come across them. So more or less, all of them are considered medical devices. So any material that goes in making the medical device, we call it as a biomaterial. But then there are certain requirements I will explain in the subsequent part of this lecture. So below there are some nice images. You have different commonly used medical devices in healthcare. And that's just a small few number. There are thousands of them. So I just give you some examples for you, just to relate to what we are talking about. Okay. Now, Dr. Lim, last time she actually gave a definition like this, which is a very detailed, legalistic definition. And she referred to US FDA, Food and Drug Administration definition. These definitions requires a lot of discussions among the experts. And finally, they agree to have this commonly acceptable definition. So why I'm showing here as a second slide, first I given my simple definition, but I'm now showing you the actual definition. The reason is, if you ever think of, say, going into business, you want to sell a medical device, and you are importing from some company from overseas, but you want to sell in Singapore, you must read this one carefully. And they have to comply with this definition. If they don't comply, then you would be in trouble. If something goes wrong, they take you to the court or there's a lawsuit, you have to defend yourself because you are liable, because you are importing and selling, distributing in Singapore. So this is mostly from the point of what are the rights in terms of the medical device suppliers, even the manufacturers or distributors, and sometimes also the users. Sometimes users has issue with respect to the medical devices, and they can complain. The users are not only the patients, even the clinicians. Sometimes clinicians may have a problem with the device because it's not designed properly and it is not meant for that, but is supplied to them. And most of the time, hospitals, they have a centralized procurement. They just procure a lot of these devices in bulk. So much later, they will start using it. Recently, I think a few years ago in Singapore, there was a case. Normally, when people get older, you have a hip and knee problems, they wear out. So one patient, maybe in his 70s, he got his hip joint replaced. Then he had a constant pain. And then this hip joint, I think I'll show you probably, it's there in a previous slide. This one, this is basically what we call a femoral, this is a femoral component that goes this part of the long bone. And this is the joint, basically, this one. That's what we call femoral head. And then you have a socket. So because this person is old, his bone is already very weak. It's porous. So somehow the doctors fixed it in Singapore. And every day the wife actually has to push it back into his body. It comes out, literally. And so they fixed it, but it just comes out. Of course, the tissue is still there, but you can see it outside. It's like outside his bone structure. So he has severe problems. And then there was an investigation. They figured out they use a medical device that is actually recalled by the manufacturer. The manufacturer actually recalled it, but this is already in the store. And then when the operation is needed, they just used it. But medical device manufacturer recalled it. There are some problems with the design. So now you see then the doctor obviously worried. Then he has to consult what is his limits. Is he within the limit or is outside? Such situations, we have to go by the definitions, standards, and regulation. So that's why I show that Dr. Joanne Lim's definition, which is the most precise and accurate, that's the one. So now if you read it, it's written in a very legalistic way. That's how it is, Any regulatory documents will be written that way. Sometimes they're quite boring, challenging to understand because they write it such a way. But that's what it is. So it's basically an instrument, apparatus, implement, machine, contrivance, implant, in vitro reagent, or other similar or related articles, including a component, a part which is recognized, official formulae, and intended for use and diagnosis of disease and other conditions, or in the cure, mitigation, treatment, prevention of diseases in man. So here there is actually word animals also, but I'm not quite sure. But I think we stick to the human beings. I wouldn't was use the word man. It should be human. Anyway, so somebody wrote some time ago, that was it is. So intended to affect the structure or function of the person, basically. So as long as that device has an influence on the structure and function of the tissue, cells, or organs, that's where we say it is a device meant for healthcare outcome. So that's basically the definition. So now this definition, it depends, let's say different countries, sometimes they may modify based on their national requirements. Sometimes they adopt whatever is given in the US FDA. It depends. So some countries, they would further align those definitions to their own need. So let's say you are practicing or somehow involved in the medical device business or healthcare business. You have to read the definitions in that country. And then you basically figure out you are doing things according to the definitions and regulations. Yeah? Okay. So then you have a very simple, rudimentary hospitals. I wouldn't want to name the country, but I visited these places. I visit hospital quite regularly because we work on healthcare research. So I visit them for our research. So this is not nearby. It's a very far away country. I had to take a 12-hour flight and you get there. It's amazing. You know, the patient volumes are huge, but the hospitals are very like scarce, very limited amount of resources. But nevertheless, they still have a medical device. So what you see there, a variety of medical devices. Then you could also get to a much more device intensive, instrument intensive hospitals, in this case, a surgery room. They're actually doing a transplant between husband and wife. I think husband probably been drinking too much and he has a problem with the liver and the wife is donating. So it's a live transplant that's going on from the wife to the husband. So they're basically two operation theaters and they're doing the live transplant. And you could actually see on the monitor image. The main point here is you could see the amount of biomaterials, amount of medical devices needed for a successful surgical procedure. So surgical procedure depends on the expertise of the clinician or the surgeon, also how well this particular device is actually made and designed. So when you start going into the choosing medical devices for a particular treatment, you will see there are lots of options, many. So that's where you need a bit of knowledge, and then you make a best possible decision. So for every disease, there are options. So I want you to look at this slide. This is a slide, basically, Singapore scenario. So Singapore medical device industry began in early 2000. I was founding director of NUS Bioengineering. I established it. And later on, current NUS Biomedical Engineering department came from that one. So we're quite happy that we started that in year 2000, at that time, 2001. So since then, we have 400 med tech companies, 25 R&D centers, 16,000 skilled labor, and 50 companies have a regional headquarters in Singapore, and 35 manufacturing plants. So it's good. It's I think it's a precision manufacturing, high value add manufacturing. So having this capability in Singapore is absolutely very necessary. Any advanced country or high income country would have to have such capabilities. And then, of course, Singapore says, according to the Economic Development Board, 9 in 10 genes chips for bioassays, they're made in Singapore. Half of the world's mass spectroscopy systems, 1 in 5 cardiovascular related devices, 1 in 10 contact lenses, and 1 in 7 hearing aids. They're made in Singapore for the global consumption. You know, the world is 8 billion people, so it's not a small number. And that population is increasing. Almost every human being are accessing healthcare. So that means, in my opinion, all other industries will have up and down, but medical device industry is always a sunshine industry because humans need them. So that's basically the idea. So last time, Dr. Lim told you in a very very detailed, complex way. But I think best way to understand is that all these medical devices essentially grouped into three classes based on what we call the risk based on the risk. So for what is the risk? If the device you use for healthcare, something goes wrong, the risk to the patient is considered as the a benchmark. If it is highly risky to the patient because something goes wrong, the life is threatening, then we call them class 3 devices. See, if it is a low risk, that means it is not really life threatening, but it is not also at the same time not good for the patient. In case something goes wrong, we classify them as class 1 device. So something in between moderate risk or medium risk, we call it as a class 2 device. So basically, the whole world, the devices are class 1, class 2, class 3. But countries like Singapore, European Union, Japan, Australia, they subcategorize class 2 into class 2A and class 2B. It's a finer definition. So now you can see, based on the country, the definitions change. So again, if you are buying or importing from one country and trying to distribute in another country, you need to make sure during the registration of the device with the National Health Agency. In Singapore, it's called HSA, Health Sciences Authority. So that is under the Minister of Health. So they are basically the one decide whether you are allowed to import and sell your medical device in Singapore. So when you submit the documents to them, you have to tell them which class is the device, what is the equivalent to Singapore. So you had to fill a lot of documentation. So my former student, Theo V. Young, he's now, he's actually an entrepreneur. He does that. So he provides that kind of service to the companies, and he's been doing it for more than 15 years. So it's a good area to be involved in case you are interested. So essentially, the higher the risk to the patient, higher the control by the regulatory authorities on the device. So that's basically a corresponding relation between the perceived risk and the control needed. So now, if you look at it, this is what the control, regulatory control, is translated into class 1, class 2, class 3 devices. If you assume all the devices would have a general control. That means they're manufactured in a good condition, what we call GMP, good manufacturing practice. So you cannot just make in any workshop or any company. No, it has to be made properly. So that's called good manufacturing practice. So when it is a good manufacturing practice, there are a lot of requirements, and that becomes expensive. Imagine this classroom, just they say it's a teaching classroom, but there's no tables, no chairs, nothing, no air condition. Yeah, there's a lecturer, there are students, you teach, that's it. That becomes very basic. But you really need nice chairs and comfortable things, the projection systems, and the teacher has all the nice device, whatever, the audio visual, you have everything you need to. Similarly, in the manufacturing, if you put all those things properly, that we call that as a good manufacturing practice. And because you need a good manufacturing practice, it becomes expensive. That's why medical devices are expensive, because the cost of making and the compliance required is very stringent. They do inspections, and if you don't comply, they close it down. So that's why it is expensive. So once somebody has set up the GMP facility, they make money, provided they have a business mind. The reason is for others to set up, they need more money. So whoever does it first, they already have experience, they can figure out. And then since the entry bar is higher, it's not easy for other guys to quickly come in and set up. So that's kind of some level of protection available automatically for the manufacturers. Okay, then some of them are, if it is class 1, class 2, with or without exemptions. What they mean is you don't need to do detailed preclinical studies for certain devices. Those, there's nothing new device, the same device, and you are making one more of those similar device. So that means this device is already there in the market or clinical use. You are making a similar device with no changes. Then it is called without or with exemptions. That means they can ask for exemptions. But again, if it is a class 3 device, no, you still have to comply. Only for the lower risk devices, you can ask for exemptions from the regulatory authority, what kind of preclinical tests you must do, and provide all that data. That data is huge, at least runs thousands of pages, not 1 or 2 pages. Each time you need to submit a dossier along with the device for seeking an approval, requests lots of information, thousands of pages. Yeah. And then special controls, especially if you're a class 3 device, this special control requires a lot more preclinical testing. That means before it is actually put in the human beings, you have to go through an extensive protocol of testing them. For example, a device you design for, say, helping a heart patient, what happens now is that has to be tested in volunteers. So volunteers have to be recruited properly. That means a hospital should get involved. The hospital cannot get involved automatically. They have to have an institutional review board, and that comprises a panel of experts other than the clinician who is going to do the testing and the patient. So they had to review the whole procedure, and they had to agree with your protocol. That's institutional review boards. Not only that, you need to make sure the patients are insured if something goes wrong. And similarly, the costs are taken care because of the clinical trial. So you have all the resources involved. And you also have a way how to analyze the data, interpret the data, present the data. So all that is required. So this is an expectation when you are making a class 3 device. So that's why the requirements, special controls are very, very stringent. And also they require a pre-market approval. That means the regulatory authority must give you the approval. Without that, you actually cannot supply in the market. So that's basically how it works. So far, we basically talked about the medical device. But this course is about materials in medicine, which is basically biomaterial. So the definition of biomaterial is a material intended to interface with the biological systems, that is our human body systems, to evaluate, to treat, to augment, or replace any tissue, organ, or function of the human body. So that's basically the definition of a biomaterial. So that's the definition of the biomaterial, but the type of biomaterial changes based on the chemistry. So in this case, metal is a metal, it's metallic ions. So they are basically, we call them metals. Whereas some of them are organic, we call them polymers. Some of them are ceramic, yeah? So it has a very strong bonds inside the atomic structures, we call them ceramics. And in this case, you could see dental crown is a ceramic material. SiO2, silicon SiO2 is a ceramic material. Al2O3, aluminum oxide, titanium oxide, TiO2, they're all ceramics, yeah? So then you have composites, which is a combination of mostly polymers and maybe some metal oxides or ceramics. So you mix polymer and a ceramic, you make it as a composite. An example is the dental fillers, especially you go to the dentist, if you have a cavity, mostly he will clean the cavity, and then what they call, they do the deburring. They use the small drill and they clean that cavity, and then he fills the cavity with the composite resin, which is basically a combination of polymer and some ceramic particles. So that sets in, and then you have a teeth which is restored, and you are back to your normal life with probably no pain. Okay, so those are the things. So when you are looking for biomaterials in medicine, there are 3 important aspects. One is safety of the patient. They don't really care whether your company is profitable or not, whether you work so hard and make the device. It doesn't matter. They don't care. You can say all you want, they don't care. All they care is the patient, is it safe to the patient? That's it. If it is not safe to the patient, any story you tell in convincing them, it is not acceptable. Period. That's it. You can say, I worked so hard and 10 years of my life I wasted. I designed this one. Please accept it. No, they don't care. It has to be safety of the patient. It's a must. Second one, after the safety, device performance. That means, is it doing the job it is supposed to do? So if you say you make a tube, it is supposed to help restoring the blood flow. Is that doing the job? And that's it. So as long as the blood flow is restored and you claim it's a blood vessel, you made it as a device, as long as it does its function, that's fine. Like, for example, you made a contact lens, yeah, it is meant to correct the vision. Has it done its job? And that's it. So that is basically the device performance. But there is one more, most important one is effectiveness. Normally, effectiveness of a device comes after many, many years of experience. That means somebody is using the device, it's working, it's fine, it is reasonably safe to the patient, and also it is doing its job. But effectiveness is not understood until many thousands of patients and many years of experience. Then you really know, is this really effective, right? So sometimes people come up with the new drug delivery methods, and then after 10 years, they say it is not working because the drug is actually not reaching to the place it is supposed to go. It is not going. Yeah, so they say it's not working. So they find out the effectiveness is not there. Similarly, those implants, I said hip implants, they were recalled back because after 10 years, they understood there are problems. There are systemic problems. So hip is fine, but when you do the hip joint, the wear out particles from the device, they slowly circulate into the human system, human body. They go and deposit somewhere else, what we call as synovial joints. And then they accumulate and they create a problem there. So that problem will come much later. Then suddenly you discover that device is not working properly. So that's where we call it effectiveness of the device. So that normally after a period of time, we would know is it really effective. So these are three important criteria with respect to the medical device. So now we can go as a very exhaustive list, and this just give you a bullet, some kind of key points, but there's many other factors. So what are the factors that influence the success of a medical device is, first of all, material selection. So you have to select the material most appropriately. For example, you are dealing with a soft tissue, you don't want to choose a material that's very, very hard. So you might want to choose a material which is similar in terms of stiffness and elastic modulus. So that's basically one example. Surface characteristics. And if the surface is not appropriate, you would actually realize later on through the video, that would influence the success or failure of the implant. The material is good, that's fine, but the surface is not good. Maybe the roughness of the surface or certain chemical structure on the surface has a negative influence on the success of the medical device. Similarly, implant design, sometimes the design has a very sharp edges, and that may not be actually viable in terms of, say, for example, the local host tissue get damaged. So the implant design is very important. We can go through a lot of examples. For example, a few years ago, one clinician told me there's a new company that designed a very nice, touch button panel for him to do the automatic dispensation of maybe insulin or some other delivery system. Then what he realized, he has 25 years of experience, but he found this device is problematic for him because his fingers are thicker, whereas these buttons on that device are very close. So he's pressing, let's say I want to inject 0.5 grams, but he's actually injecting 2 grams. That's the next button, because his finger is thicker. The guy who designed it, he did not imagine, there is different sizes and shapes of the fingers, just fingers alone. So that's a wrong design, right? So then he has to give a feedback and then some issues like that. So just an example. Biocompatibility, I'm going to talk to that in more detail. Then the sterilization, very important. Any device that should be sterilizable. That means any bacteria, microorganisms that are there that need to be killed by sterilization techniques. There are many types, gamma rays, X-rays. They use specific gases. They also use steam for different ways of sterilization. And then the clinical procedure. The implant is designed very well, but the doctor is not following a good clinical procedure. Then the also implant can fail. That means the surgeon's ability also comes into the picture. Everything is fine, but the patient is also not responsible. So they've been asked to, use it properly. They don't. For example, when people have a prostate cancer, one other treatment is brachytherapy. So they insert rods through which they actually do the radiation. And because the patient has to go through multiple times radiation, the rods are left inside, and then a person is asked to go home and come back again. So the patient really had to take care. The rods don't shift too much. Also, at the same time, keep the rods free from infections. Otherwise, again, they'll create a problem. So that is very much depends on how responsible is the patient. So patient need to be a bit of responsible and knowledgeable with whatever advice is given by the doctor. So again, everything goes well. Sometimes patient's issue also can make the device go wrong. And then, of course, try to make the device as error-free as possible so that anybody can know intuitively how to use it in terms of following the procedure. So these are some of the requirements, but there are many, many other requirements. So an important aspect of this one is biocompatibility. So all biomaterials, we normally say it should have a biocompatibility. It's defined as its ability to perform an appropriate host response in a specific application. Tissue tolerance, mild rejection, or fibrous encapsulation are evidence of biocompatibility. And biocompatibility evaluation involves chemical toxicity, bacterial toxicity, mechanical irritation, and many others. I will show the list. So before I can go into further, there are 2 videos interesting for you to understand. So I will play them. And I don't know whether the guys online can see it, but I suggest online guys can watch at their own time. Okay, let's play. Okay, the first one is foreign body reaction. Let me stop share and then share again. Let us see. Okay, share. That's the one. Yeah, okay. Hopefully they can see. Okay, I will play it now.
Vinyl difluoride and polypropylene on foreign body reaction, a comparison. Have you ever wondered what actually happens when you, the surgeon, implant a mesh? How does the tissue react to the polymer and the structure? Imagine you are stood on the surface of the mesh and can observe what happens here just a few milliseconds after the latter's implantation. Almost immediately after implantation of the mesh, autologous proteins such as albumin and fibrinogen are absorbed on the surface. The changes to and degeneration of the proteins represents a strong immunological stimulus, in particular for the macrophage, which can be found at the interface just a few minutes after implantation. The macrophage is activated as a result of the contact with the proteins and, in turn, releases messengers which serve to initiate the foreign body reaction. Ever more macrophages are attracted chemotactically to the site, where they form the early foreign body granuloma. It takes only a few hours for the mesh to be enveloped and fully covered by the macrophages. During release of the messengers, other important cells are also attracted, in particular fibroblasts, which are responsible for the cicatricial envelopment of the mesh. Even on day one, many fibroblasts are present, which form an early parafilamentary scar. Both T lymphocytes and polymorphonuclear granulocytes are present, which disappear as the granuloma ages. As the process continues, the number of cells in the granuloma increases, and after 1 week, an early foreign body granuloma can be recognized with its typical layering. In the interior, the macrophage layer, and outside the fibrous or cicatricial capsule comprising collagen fibers and a great number of fibroblasts. The foreign body granuloma now starts to age, and there is increased vascularization, a process which is evident after only 2 weeks. Endothelial cells firstly form simple endothelial tubes, which subsequently develop into capillaries. Here, good dense vascularization is proof of the optimal integration of the material in the implant bed. The extent of the foreign body reaction and the extent to which the granuloma envelops the individual mesh fibers essentially depends on the polymer used. The intensity and type of macrophage activity on the surface is closely correlated with the thickness and diameter of the fibrous capsule in the outer granuloma area, the parameter which is of particular importance for the subsequent functioning of the mesh. The foreign body reaction of the PVDF is minimal. Due to the lesser stimulation of the macrophages during implant healing, this polymer results in a significantly smaller granuloma width and fibrous capsule development. In contrast, we have the PP. Here, both the macrophage layer and the peripheral connective tissue capsule are more pronounced. Let's now leave the surface of the mesh and take another look at the implant as a whole from above. Following implantation, the mesh is very quickly covered with proteins, which form the foreign body granuloma and a cicatricial covering around the mesh. The scar capsule is shown in purple here. And again as a cross-section, inside the mesh, then the foreign body granuloma and the outer scar capsule. The comparison of PVDF and PP shows that, given the same suture thickness and pore size with PVDF, the scar capsule is significantly smaller. With PP, there is increased bridging due to scar tissue formation, which, in part, completely fills the pores of the mesh. For our PP mesh, this means that the pore structure is essentially lost and that there is a more extensive cicatricial covering around it. In contrast, the PVDF mesh is shown on the left here. For the most part, the pore structure has been retained. Healing of the mesh implant into the abdominal wall with only minimal cicatricial capsule formation is of particular importance to patients. Minimal foreign body reaction and scar formation lead to optimal integration of the implant, and as a result of the reduced level of scarring, the flexibility of the mesh is retained. For your patients, the reduced connective tissue induction of PVDF equates to retained flexibility of the abdominal wall, as well as optimal wearing comfort, with significantly reduced foreign body sensation.
Let me play another one, and then I will start my lecture again.
Usually, every time you have a cut or bruise, your blood clots to stop the bleeding. Clotting is a complex process, and there are four main things that make it possible: platelets or cell fragments, clotting factors, which are special proteins, fibrin, which is a protein mesh made up of a special kind of clotting factor, and finally, other cells, like red and white blood cells. Because clot formation is so important, platelets and clotting factors are always available, floating around in your blood. Now, let's talk about the four steps involved with forming a blood clot. When there is a tear in a blood vessel, the first thing that happens is that the nearby platelets are activated and become sticky. They start sticking to each other and to the sides of the hole in the blood vessel. For small holes, enough platelets usually stick together to form a temporary plug. But a platelet plug isn't strong enough to block the opening for long, so it must be reinforced with other materials. Otherwise, the blood flowing past the hole could wash the plug away. These other materials are called clotting factors. As the clotting factors float by the tear, they become activated or turned on and add themselves to the platelet plug. A special kind of clotting factor can weave itself together with others of the same kind and form a web of fibrous tissue called fibrin. This web acts like glue and holds the platelets and the other clotting factors together, creating a blood clot. Other cells, like red and white blood cells, can also get caught in this web and help reinforce the clot. It quickly becomes stronger and starts to pull the edges of the hole in the blood vessel together. After the clot has formed, it stays in the hole until the tissue is repaired. When the clot is no longer needed, your body dissolves it.
When you see this green arrow over here, you buy. When you
Okay, basically through that two videos, I showed the importance of material type. In this case, polymer, but they have two different polymers, polypropylene, PVDF, another polymer, and they have different chemistry, different interaction with the host cells and tissues. So you can see the foreign body reaction is different. And to understand that, look at this one. So you have a biomaterial, for example, maybe it's used for surgical purposes, and that's coming in contact with the blood, which is circulating in the body. So the blood contains many things, and one of that is proteins, what they call, actually blood has lots of proteins and many cells, they have many types. And normally you know them as red blood cells, white blood cells, but in addition to that, many other proteins that are actually constantly circulating in our body from the heart. So because you have those things, when there is a cut in your hand or in your skin or inside your body because of surgery, the foreign body reaction takes place. And in this case, if you are implanting a biomaterial or a medical device, to implant that one also, you are disturbing the tissue. And it requires that it promotes a foreign body reaction. So that's a must. So a foreign body reaction involves initially protein adsorption on the surface. Then after that, you have cells like platelets and monocytes. They try to deposit on the biomaterial surface. Then after that, there are the times there are other cytokines. These are basically biomolecules released by the cells in that particular environment. There are many types of cytokines, and they need to be released because they're trying to protect you from the damage. And then after that, there is a tissue repair. There is another cells, what we call fibroblasts. And they are the one form a fence around the implant, because body recognizes biomaterial or medical device as a foreign body. It's not yours. So when a body recognizes that, it actually has this reaction. as long as that reaction is mild and within the acceptable limit, it's a good reaction. And so what we are showing here is how a typical foreign body reaction eventually results in a formation of a fibrous capsule around the implant. So that's basically what happens. And then the same, if you're also looking at, let's say there is a wound in the skin that is either unintentionally you have a cut or surgical procedure that involves cutting the tissue. So that has to trigger the wound healing process. Otherwise, the blood is coming out and you lose the blood and eventually you faint and eventually fatality may happen. In fact, diabetes patients, one of the problem is the clotting doesn't happen because of the change in the cellular system and the way whole system is working. Much more difficult for a diabetes patients for the blood clotting. So that's why doctors actually ask you, do you have any other conditions before they treat you? Yeah. So a normal healthy person with a certain, say, cut in the soft tissues, you would have this kind of reaction. So first is the inflammation, followed by re-epithelialization and proliferation. Finally, it is a remodeling. So remodeling is the formation of the fibroblast-mediated fibrous tissue formation. So this is the standard procedure, and it is to be expected when you have a good biomaterial, very well-designed implant. So biocompatibility involves this kind of processes. Yeah. So now to assess the biocompatibility, these are more detailed. There is an ISO standard. It's called ISO 10993. So this is the gold standard. Basically, if you are a biomaterial scientist, and you must know this, it's called ISO 10993. So it's a series of standards. Even though it is one number, they have sub numbers, and it's a series of standards that detail various biocompatibility tests needed for medical grade materials and devices. So when any regulatory authority giving you the permission to market a medical device, they are not certifying the material. They are actually certifying the medical device. Of course, there is no device without the material, but all the regulatory approvals are given to you for the device, not the biomaterial. Yeah. So now in this case, there are lots of tests, and we're not going to go through all the tests, but for you to understand that many series of steps, tests are involved. For example, cytotoxic, sensitization, irritation, hematocompatibility, and here is the numbers, cross numbers, 10993-5, 10, and so on. So the experts in a medical device field, they will advise you which tests are to be done based on the device, based on the treatment you're going to use that device for. So you do not need to do all the tests. So they will advise you which test to be done. So normally, if I'm going to drill down deeply in this area, I would give you one device, and then I'll ask you, can you decide which test to be done and why? So we can ask a question like that. But this year, we're not going to do that. This year, we change our continuous assessment focus. So I would not ask you that. But if you go like 2-3 years ago, I was asking students, tell me, this is the device, what kind of tests we need to do and why. So that's how you can deepen your knowledge and also what happens in the medical device industry. So now I know many of you are mechanical engineering students like me. I'm a mechanical engineering person. So when we are talking about the biocompatibility and design, eventually we have to test the material, we have to test the device. Tests are many different types. It depends what kind of tests. But I'm just giving a simple, the most basic mechanical test, which is a tensile test. Yeah. And I realized last time some students from other background disciplines, they are not familiar with this methodology. So just let me explain very simple way. This is a simple tensile test. That means you want to know what is the failure strength of the material. What is the stiffness? We normally call elastic modulus. And also we want to know what is how much you can stretch the material, what we call strain, before it fails. These are simple 3 parameters we find information, properties of the material by simple tensile tests. Then you do compression, you can do fatigue, creep, and many other tests. But we're not going to go through all of them. But the main point I want to basically say to you is if you look at it in the standard, they give you how to make the specimen sample of the biomaterial and how to test it. All the methodology is given. And when you test as per the procedure, if it is a polymer, you would get a stress strain curve like that. If it is a ceramic, you would get it like that. And a metal is here and a composite would be in between. So now you can understand from that, based on the four major types of materials, the general behavior of the materials is different. So again, within metals, polymers, ceramics, composites, there are thousands of materials. There's no way we can cover all those materials one by one. So this is for you to just get a broad idea. When you say polymer, you should, in your mind, polymer is, that is a stress strain curve. If it is a ceramic, that's what it is. Metal is like that. So if you look at your teeth, that should have a mechanical behavior like this, ceramic, your teeth. You are looking at your skin, it would have a polymer type of behavior. So soft tissues, generally polymer type, ceramics are hard tissues, metals are in between, composites are in between. So that's kind of intuitively, you get a basic concept. This is I would say 101 of a material knowledge. But those who have a basic degree, they should have much more knowledge. So, but I'm mindful not everyone has materials background. Some of you are completely different background. So I'm giving a most simple test that we normally use for determining the property. So then we have, how we find certain properties. In this case, the initial curve of the stress strain curve, the initial part, and we look at the slope and we call that as an elastic modulus. And then there is a tensile strength is the end point which the sample fails, and the strain at which it is failing, we call it as a failure strain. So these are like simple parameters, characteristics of a material. So we put that aside. Now using that information, how do I compare different biomaterials? So here, materials. So you have metal. When I say metal, is actually metal alloys, and titanium 6Al4V, this is titanium alloy, chromium alloy, stainless steel. They are the one used for making metallic implants like bone screws and bone plates and joints. Similarly, you have polymers like PTFE, PVDF, polypropylene, and so on. And then ceramics. And now if you look at it, see a polymer has a tensile strength and elastic modulus in that range, whereas the metals have a much higher number. And a ceramic obviously has the highest stiffness or elastic modulus, which is almost 380 gigapascals. Whereas a mechanical strength or tensile strength for metals is very high. It's anywhere from 600 to 2000 megapascals. And the polymers are weaker, but the polymers have their own function in a biomedical application. So now if you compare that with the cortical bone, it's basically the hardest part of your body. There are two things, the enamel on the teeth and the bone. These are the hardest part in your body. And if you look at the cortical bone, that would have a tensile strength of 50 to 170 megapascals. You can see that's much higher than polymer, but it's actually lower than metals and ceramics. Similarly, elastic modulus of a cortical bone, much lower than your ceramic, much higher than polymers, and somewhat much lower than metal alloys. So you could see 4 broad categories of materials and how they relate to the actual bone or the human tissue. Now, to make things more complicated, here it is. A bone, it looks very simple, but it's very complicated. A typical bone, this is a femur bone, so it's in your thigh, it's a femur bone. It would have a complex hierarchical geometry. So if you start looking at that particular bone very closely, you will find the fibers which are made of collagen, and it also has the hydroxyapatite crystals, which are ceramic. And these things are interlaced like chain, and they are twisted and turned and made into hierarchical structures. So your bone evolves with time. Why this evolves so complicated is because as you start growing from your zero, as you grow, your bone starts remodeling all the time based on your physical exercises, your mobility, the way you live, your lifestyle. So that's actually remodels your bone. That's why some people have a much stronger bone, more dense bone than other people. It depends. Yeah. So generally, everyone would have that kind of hierarchical structure. So now if you see the bone, you have a cortical bone, and you have this longitudinal direction and transverse direction. That means the properties in this direction and properties in this direction, they're not the same. So the material is directional. The properties of any tissue is directional. So then you have a soft tissues which are which has a lower modulus and tensile strength compared to the hard tissues like a bone and so on. So if you look at the leftmost here comparison, I like this one very much. Here we compare skin, tendon, bone, and also the brain. So look at the brain. The softest part of your body is actually the brain. So you have the skull. Of course, you can see my skull. It's very, very, tough. But inside the brain is very, very soft. It's gooey. Yeah. So that's why I won't go into the detail. But when people have, high-speed crashes, the brain actually decelerates at a particular gravity. And if you cross certain number, that damages the brain quite a bit. So that's how when we design Formula One racing cars, we actually design them so that deceleration due to accident is not too much on your brain. So that's how this knowledge goes in terms of designing the products which we use for fun or anything else. Okay, so we can go on a lot of details. We write lots of papers and books on these things. But for you to get an idea how complex human body and each tissue and how they compare with each other. Okay, so to make it even more complicated for you, look at the hand, your own hand or my own hand. So it is not that simple. So the hand basically have muscles, it has a bone, it has blood vessels, and each of them, if you start looking inside, the tissue structures are different because they have different types of cells. And each cell has a different function. So sometimes all these cells, they function well, sometimes they don't function well. That's where the diseases come or the complications. And then sometimes you intentionally create damage. For example, you meet with an accident or you fight with people and then you have cuts and fractures and then there is a breakage and these need to be solved. Some are genetic, some people have certain issues. So fundamentally such a complex evolved structures can go wrong. So nothing can be taken granted. And each one of you, I keep saying, very different. That's because of the complexity. No two individuals are similar because of this complexity. So the millions and billions of cells behind these tissues and structures and organs. Yeah. Okay. So then all these whether it is bone or muscle or skin or blood vessel, all these things are essentially come from what we call stem cells. These are the primary cells. So from the stem cells, they differentiate and they form different types of cells based on the tissue or the organ. For example, fat cell becomes fat, bone cell forms the bone, blood cells supplies the oxygen, and nerve cells basically helps you to understand senses, make sense of it, send signals, and receive the signals, and interpret the signals. And then you have epithelial cells, it covers your body, protects you from the harmful microorganisms, and you have immune cells, which are behind the scene protecting you. So, and so on, you have many things, yeah? So in summary of this particular course, basically of this today's lecture, global market for medical devices is about $1 trillion. It's not small. And the global market for biomaterials to make the devices would be about $200 billion. And there are lots of those devices, yeah? And almost right now, every part of the human body, there is some device for supporting, augmenting, treating, or helping. It's plenty. And then you can see with all these medical advancements, we still have a challenge is there is a gap between lifespan and healthy span is about 10 years. So according to Singapore Ministry of Health, as well as other like World Health Organization, before a person dies, last 1 year, 5 years, or 10 years is extremely crucial. So my general advice for you is the moment you get yourself a job, get yourself a good life insurance and health insurance. It's necessary. Very expensive nowadays. Why? Because you don't know. So the take-home message from my lecture is the body is complex. You can be as good as you can be, but it's very complicated. It's a random process and nothing to do with you. It happens. So once you get your job, make sure you have a health coverage. Extremely important. Yeah. And then the last 5, 10 years or 1 year is most challenging. So if you have elderly people at your home, grandparents and whatever, make sure they have a good coverage. Otherwise, it's quite a challenging for them. This is all very expensive. So this is hospitals and things. I'm not promoting my own book, but in case somebody is really, really interested in this one, you don't need to read it. But if you think medical device is an area, you want to build your career, you had to start with this one, regulations, standards, and also classification, protocols and things. So, but you don't need to even look at that if you are just going to go through this course and you're not actually interested in pursuing further, yeah? So that's just a basic idea. So now I will stop recording because we have a part 2 today. Okay. Okay, guys, let's go through the CA guidelines. Over the last 3 weeks, we have done some kind of changes to the CA. When we started with it was supposed to be 1 and also should be 10,000 words, but we changed it to 2. And then initially it was 3,500 words each. Now we changed to 3,000 each. And also I had different definitions like critical illness, terminal illness, and unmet clinical needs. But I changed it to more simplified version. Any disease is fine. And then there's more clear definition in terms of nanotechnology in medical field. Yeah. So main point is we already reached first 4 weeks of this course. So everything you see today, that's final. So there won't be any change. So if you plan to start working on your CA, you can start from today. Yeah. So there won't be any more changes. I have taken all the feedback. I have refined as much as we can, but I cannot change all the time because some people start working and we cannot change the guideline anymore. Yeah. Okay, so this is the final version. So if I go through it, so it's 100% CA. I'm just saying it again, there is no final exam. Your CA reports are the final. There won't be any final exam for this course. The next one is we have equal weightage in terms of marks. CA1 is about diseases and treatments. CA2 is medical applications or nanomaterials. Both has basically 50 marks and 50 marks. That's right. So if you choose to submit only one, that means you're only aiming for 50 marks out of 100. So it's better you do both of them. Otherwise, you straight away lose 50%. Yeah, remember that. So it's 50% and 50%. And generally, we look for the quality of writing, accuracy of the information, depth of analysis, and clarity of reasoning. That's basically the weightage. So this comprise 100% of both of them. So we will look at, for each case, I look at that kind of distribution. Again, no matter which slide, whatever, wherever you saw before, forget about all of them. Total length of each report, CA1 or CA2, each one should be about 3,000 different words, including references. That means you don't want to have too many references. I suggest you have about 20 to 30 references, or maybe 40 references. It's okay. You don't need a lot of references, okay? And if you are ambitious and you want to exclude the references, I don't mind, up to you. But I need to give a guideline. So I just put that as a guideline as including references. But some guys say, no, Prof, I want to put references, exclude them, up to you. I wouldn't give any special weightage, whether you include or not include in a 3,000 words, it's fine. The most important is the similarity level. Plagiarism check is a must and should be near 0. It's also impossible to achieve, but I just say it because now with generative AI and all those things that you're going to use, because I wouldn't know whether you are using or not. The thing is very likely you might be plagiarizing a lot. And first thing, NUS would not like you to do plagiarism. Obviously, that's not a good thing. Even more important is in your future, if you ever plagiarize before, that will haunt you. I'm very sure about that. So no matter what you do in your life, you have to take plagiarism as the most serious thing because that affects your professional standing, no matter what you do, because people can accuse you on plagiarism. So, and then nobody can help you because it's an evidence is there. So it's a very terrible thing nowadays happening because unintentionally people plagiarize and it's giving a lot of headaches. Like NUS now ask every researcher go through special training about these things, you know. Every researcher has mandatory, they had to do it. It's not because NUS, it's just worldwide, you know, software development is so good. It is giving other problems. So bottom line is try and bring it as less as possible. The general guidelines about 3%, 2%, but I put near 0 so that we'll see how it happens. Yeah. Okay. But it cannot be 10%. That'll be too much. So somewhere near 0, I put it intentionally, I'm vague. But if it is 2%, 3%, I can still accept it. Okay. And then cite primarily papers from Q1 journals of SJR, and that's a link. You click on it, and any reference you find, take the name of the journal and check on SJR. It's a Q1. As long as it's quartile 1, that is a good reference. So that's the one way for you to know it's a good reference. And the total references for each to be limited to 30. I would recommend maybe even 20. 20 to 30 is good in each report. And combine CA1 and CA2 as one single report and submit it. And make sure you have your matriculation number, your registered name as per the NUS. And then I will create a folder later on, and you can submit that in a CA folder. So make sure there is a matriculation number and your name, because otherwise it's going to be very difficult for me. We have 231 students. If I can't figure out your whatever your report, I had to search all the reports. Not easy, it's quite difficult. So make sure put your name and your matriculation number as in the file, and then I know That's yours. So we would verify because when I enter the marks, I have to cross-check I'm entering the marks against the right name. So sometimes names are very similar and I don't want to make a mistake. So help me not to make the mistake by making sure you put your matriculation number and the name and submit it. Yeah. So your report should have those names. So name of the file should have those two. That's most important. You submit. And there is a late submission penalty if you submit late. It is required you do an in-depth literature review. You have to draw your own schematics, infographics, and tables. Somebody asked me what kind of software. Previously, I used to give some softwares, like I list them, but I find now too many softwares. I can't even follow how many softwares are available. So I leave it to you. You decide whatever software you want to use, please go ahead and use them. No problem. Last year, actually, I listed which softwares you can use, but this year, I'm not giving any software names. It's up to you. And you're encouraged to use the descriptive captions. Normally, when you have a figure, schematic, infographic, or a table, the caption should be as descriptive as possible. It's good so that if someone looks at your illustration, they understand what you want to convey. So that's the main point you may want to note. So that's it. And then when it comes to CA1, you go to this website. This is a Singapore National University Hospital website. You click on it, you will have hundreds of diseases there, plenty. So which one to choose is up to you. I don't have a preference. You have your own preference, whatever reason. Maybe you're more familiar, you're doing research, or your family member has some kind of relation to that kind of disease, and then you want to understand more, or you already understand so much, you want to write about it, that's fine. Yeah. So basically that, and that's more like an example case. And then you should basically, for the CA1, you write what disease you chose and the causes of the disease and symptoms and the global trends, and then what are the current diagnosis and treatment options and what kind of devices and materials are used, and your critical perspective reasons. For example, you think those treatments are not satisfactory, better treatments are needed, or some other issues with respect to the treatments, you can write about that. And then foresight, that means as a graduate student of National University of Singapore, this is supposed to be top 10 universities in the world. So obviously, you are expected to think for the future, right? You're not in some unknown university. NUS is a top 10. Top 10 means you are a thought leader, right? So if you're a thought leader means you have to think. So you have to think, that's it, yeah? Because you are a student of this university. And I don't want to say I'm teaching you, that'll be too much to say. But I'm teaching because I'm also a very well-known guy in this area. So it's expected that you do something about it, yeah? Okay, so that's basically, but no pressure, don't take it too strong. I just want to try. If you find this too hard, it's okay, don't worry, but try it. Okay. And similarly, the medical applications of nanomaterials, this is the, I just give a Wikipedia definition. Basically, nanomaterial is basically any material with a single dimension less than 100 nanometers. That's basically the broad definition. So a material with a certain smallest dimension, it's called nanomaterial, and it has a higher, better functionality when you compare to the same material in a macro way. So a material, when you reduce the size to a finer size, they have enhanced functional properties. And we make use of it and we exploit them in an application. So in this case, what kind of applications are there for nanomaterials? And I chose nanomaterials because that's the most cutting edge area in terms of materials. It's been around for the last 20 years. And again, actually, I'm the pioneer of nanomaterials in Singapore. I established the NUS Nanoscience Nanotechnology Initiative 23 years ago. So we started promoting nanotechnology in Singapore year 2000. And of course, there are companies and a good situation in the economic contribution. So before that, there is no such industry in Singapore. So that's why I'm a little bit passionate about nanomaterials. And then there's kind of examples. So idea is you select a nanobiomaterial. You remember, I showed different types like metals, ceramics, polymers, composites. But then if you look for a specific biomaterial, there would be specific chemistry. Maybe it is a particular material like polyethylene glycol. It depends. What do you choose? And then that is a nanomaterial. Then you start understanding deeply about that. So you need to provide how it is actually made, production methods, characteristics, and properties, and comprehensive view of potential biomedical uses, and how actually that material is functioning as a device in that particular application, and compare and contrast performance with conventional biomaterial. That means this is the nanomaterial. Suppose it is not nanobiomaterial, it's just a biomaterial. Is there a performance difference between a normal material, nanobiomaterial? So is the nano concept has any improved functionality is what you need to talk about. And then critical response and then foresight. Again, as I said, you are in a top university. You have to think, a little bit, stretch your neurons and say something in terms of the foresight. Yeah. So that's pretty much. If you have any questions, we can go through it. But otherwise, you start working on it, and then you can ask me the questions. I'm available, and I can clear your doubts. Best is you start writing. Then you got the question, then we can answer. Otherwise, you won't be able to answer ask the right question. Yeah. So the details are already there. They are meant as a broad guideline. But you have the flexibility how you want to write your report and then present yourself. Yeah. Any questions? So from today onwards, you may want to start working on them. The reason is it takes time. Second is, nobody knows what happens tomorrow, right? Something else happens in your life or you're busy with something else. So that takes away your time and attention and mindset. So even though you have plenty of time ahead, my suggestion is you can write it. Nowadays, plenty of databases like Web of Science, Scopus, and you have, again, many other softwares. With the keywords, you will start finding information, and then you just start writing it. First, you need to read. After you read, you write in your own words, in your own sentences, and then after that, you come up with your own diagrams, illustrations. You'll be fine. It's only 3,000 words per CA report, and totally 6,000. Yeah, I think that's reasonable. I think I cannot go lower than that. I think NUS will say you must maintain standards. So I have to maintain standard. Yeah. Yeah. Okay, guys, any questions? Okay, if not, you start working on it, and then the subsequent classes, we can go through your doubts. And sometimes if you're shy, you don't have to ask openly. In the future, I'll just hang around there. You can come to me and then ask me. I can answer your question. Some people are not comfortable openly asking, but you can see me there and we can talk. So one more last one before I end today. Let me stop the share. Okay, so let me go back to this one. Okay, that's a total outline of our course. So now we are already on September 3rd, yeah? So we have today discussed the biomaterial, devices, properties, and functions, and CA1, CA2 guidelines. So next week, there is no class. So I don't see you next week. So if you don't want to come here, no need to come. Or you want to come here and do your work, it's fine. But I won't be here. So next week is your time. Start working on CA projects. Okay? next week. And then there is a one video by Dr. Bojan Lee. He's a very nice guy. He's in Switzerland. He's actually my former student, but he's now living in Switzerland for the last 15 years. And he actually works in a hospital, and then he works on the advanced therapy. So I asked him to make one video and slides. It's all there in the file with the date. You can go through his video. So he talks about stem cell treatment. It's the most advanced technology in treating diseases. And he's doing that in one of the Swiss hospital. Again, like you guys, he was my student 15 years ago. And then he decided to go to Europe, and then he's happy there. And he's quite happy to do this kind of work. So it'll be very motivating for you to just listen to his talk. So you start working, you can listen to his lecture anytime. Then I will see you again on 17th September, okay? Okay, let me stop the recording, otherwise it's too long on the system. Stop recording.